I'm just going to jump straight in and pick up where Hayu left off, talking about genome assembly, which is obviously this core bioinformatics process we've been doing for years, but it's been greatly enhanced in recent years by the emergence of long-read sequencing and also excellent assembly algorithms like Hi-Fi ASM. And as a result, it's now possible for us to generate really highly accurate and highly complete genome assemblies of plants, animals, humans, and that's just really, really fantastic. But unfortunately, it's still quite an expensive and technically challenging process. So the sort of current best practice recipe, which Hayu mentioned, sort of calls for several flow cells or smart cells of high accuracy PacBio data, several flow cells of Nanopore ultra-long read data, which is quite a finicky technique. Also some long-range techniques, so high-C, poor-C, or strand-seq. And so this means you need access to multiple sequencing platforms. It's expensive to generate all this data. It requires a lot of expertise to run all of these different techniques and then to integrate all of that data into a really high-quality assembly. And so overall, I think it's fair to say the barriers are just too high for many research labs, and there's a really clear need for us to develop ongoing solutions to improve the affordability of data production, increase the range of usable sample types, and more easily resolve the most repetitive genome regions. And so we've heard from Hayu about some of the fantastic work that his team is doing in this space, and many others working on similar things, but I'd like to add to this conversation by talking about a recent innovation from my team, which is adaptively coupled genome sequencing and assembly strategy that we've developed, which we're calling Cornetto, for reasons I don't have time to explain. So I'm just going to start off with some really basics. This is an NX plot. It's a way of visualizing a genome assembly, the contiguity or the quality of the assembly. And so on the y-axis, we're basically plotting contig sizes sorted from largest to smallest, and ideally we want every chromosome in our genome to be covered by a single contig. So the first in the T-to-T assembly that I'm showing here, the first contig is human chromosome 1, the second contig is human chromosome 2, and so on and so forth. And then on the x-axis, we're showing the cumulative assembly size growing as we add each contig, and those are sorted from largest to smallest. So that's what the T-to-T genome looks like, but for the sake of argument, if we run one PacBio smart cell and we generate a genome assembly, this is what it looks like. We don't get 23 nice complete chromosomes. We get a much more fragmented thing with lots of these shorter contigs. It's pretty good, but we know that sequencing depth is a really key consideration, and so we run a couple of extra smart cells, and that's going to improve our assembly. So you can see these lines are moving upwards and to the right. The assembly is basically getting less fragmented. But what you can also see is that there's kind of diminishing returns. Like this third smart cell didn't really add much value. Even the second smart cell didn't add all that much value. And I mean, the assembly is pretty good. We've got 90% of the genome in sort of pretty long contigs, 18 megabases or above, which used to be considered a very, very good genome assembly. But the problem is we're doing all of this sequencing, with these additional cells just to try and improve on that last 10% of the genome, which of course is the most highly repetitive, complex, mysterious regions. And so we sort of figured we're kind of wasting a lot of time and energy sequencing the whole genome again and again, deeper and deeper, and a targeted approach would sort of make a lot more sense here. We figured we could achieve this with adaptive sampling or read-until or read-fish, which is this technique you're probably all aware of, where we can program the device to recognize DNA strands during the sequencing experiment. If the strand is something you're not interested in, it reverses the charge and it spits that strand out, so you're not sequencing parts of the genome that you're not interested in. And so what we wanted to do here was basically program the device to spit out the bits of the genome that we're not interested in, which is basically the non-repetitive bits, the easy parts of the genome, or for our purposes here, the boring bits of the genome. And so this is kind of the strategy we came up with. We start off with a base assembly of sort of moderate quality, which could be generated from one PacBio smart cell or one Nanopore flow cell, for example. We take this assembly, we then label all of the regions that have sort of poor coverage, or they have low mapping quality, or they have low assembly quality. We also label any short contigs, or any region that's just close to the end of a contig. And these are basically the tricky parts of our assembly where having additional data would probably be useful. In addition, we identify any part of the assembly that's not phased, so where there's a gap between one or both of the haplotypes relative to the primary assembly, we add those, and then we aggregate together all of those regions, and then everything else, so the total remainder of the genome, we consider boring, and we then run our sequencing experiment on a new flow cell using adaptive sampling, and basically identifying any read that's originating from a boring bit, we spit it out, and so that's going to focus the data onto our challenging unsolved regions. But the cool thing is we can do this sort of dynamically. So every couple of hours, we can stop the experiment, collect all of the data, reassemble the genome. That's going to join together some of the contigs. It's going to make the assembly less fragmented. And then we can actually update our list of boring bits, and we can reload the flow cell and start the experiment again. We can sort of do this iteratively. We can run multiple flow cells if we want, and we're basically updating those boring bits to focus the experiment, focus the data production onto an increasingly small and difficult unsolved portion of the genome. And as you can see, our assembly is sort of getting better in the cold through to the hot colors as we go. So that data that I showed there was actually a hybrid assembly of PacBio data with Nanopore duplex data, but we're at London Calling, so we want to see some Nanopore-only assemblies like Ira was talking about. And so for this next experiment, we've generated a HG002 reference sample assembly using Nanopore data only. So we generate a base assembly with just standard LSK114 simplex reads, which I'm showing here. It's got 2 lines because it's a phased assembly. We have 2 separate haplotypes. That's just one flow cell of bog standard data. We then use that exact same library, run it on a second flow cell with 4 cycles of that iterative Cornetto adaptive assembly process that I showed. And as you can see, the assembly is greatly improved, and those lines are now getting pretty close to our T-to-T reference in terms of contiguity. You can also see if we align the assembled haplotypes to our T-to-T HG002 reference sequence, which we take as a truth here, that it's highly concordant. We don't have any large misassemblies. Cornetto is not buggering the assembly up and causing all sorts of artifacts, so that's great. And to kind of put this in context, we actually compare this assembly to an assembly that was published recently by the T-to-T consortium, who are real world leaders in this field. They published a paper where they were trying to do a Nanopore-only assembly, and they generated a really excellent assembly. But to do so, they used something like 10 flow cells of duplex data, 3 flow cells of ultra-long data, 3 flow cells of poor C data. So I think that's at least 16 flow cells, which is very expensive and cumbersome to do. By comparison, we use 2 flow cells, just 1 library, standard LSK114. And so I think that really is where our assembly is, where the advantages of our Cornetto process are coming into its own. The assemblies are very similar in terms of contiguity. Also, if we look at how many complete chromosomes we got sort of 27 out of a possible 46 complete chromosomes as single contigs. Their assembly had 26. Across a whole other range of standard assembly metrics, the assemblies are more or less comparable. And so we're really happy with how this is working, because we've, again, just used two flow cells, 1 library prep, standard LSK114. But beyond the fact that it's cheaper and requires less labor, the other kind of advantage here is we're using a lot less material, and that's going to open this process up of genome assembly to much more limited sample types, where you can't get bucket loads of high molecular weight DNA, or you can't do high C, or you can't do ultra long. So things like animal tissues, plant tissues. These techniques are very difficult. One material we're particularly interested in is saliva. So there's really big practical advantages to working with saliva rather than fresh blood for all sorts of applications. It's easy to collect. You just spit in a tube. You don't need a medical professional there. You do this at room temperature. You can store it at room temperature. You can post it around the world. And there's less cultural sensitivity than working with blood, which is important in some of the sort of indigenous genomics work that we do. And so, big practical advantages, but unfortunately saliva DNA, it's really hard to get lots of high molecular weight DNA, and it's often heavily contaminated with non-human material. It's not amenable to ultra-long sequencing. It's not amenable to high C or poor C. And so to our knowledge, no one's ever really generated a very highly complete genome assembly from saliva. So we set out to test if we could do this using Cornetto. We got two volunteers to spit in some tubes, and we generated assemblies, again, just using 2 nanopore flow cells, 1 standard library prep. You can see the light blue and pink lines, which are our Cornetto assemblies, are much, much better than what we can get without Cornetto, and when we get really high quality assemblies with 26 and 27 complete chromosomes respectively from our 2 donors. So we're really happy with how well that worked, and we think this is going to open up a lot of possibilities. I don't have much time left, but just to say this is a totally agnostic process. We designed it so that you don't need any prior knowledge about the genome you're assembling. We're just recognizing those features that describe an unsolved region, and that's how we choose our target regions. This means it works on, in theory, any genome. You don't need to know anything about it. So we've so far tested on a whole range of non-human vertebrates. These 3 across the top are interesting endangered Australian endemic species that we're working with with conservation groups. The other ones are fish from the Amazon. Basically, the Cornetto assemblies we get are all very highly contiguous, and if we blow up the example here of the western saw-shelled turtle, we get after Cornetto, we get 12 complete chromosomes on each haplotype, which is almost the whole genome. And so, yeah, overall it's working really well on a diverse range of species, a diverse range of sample types, and so we're really excited about it. So just to summarize, Cornetto is this adaptive integrated assembly method that we've developed, and the goal is to streamline the genome assembly process, open up new sample types, make it cheaper, human, non-human, all on the table. I like to finish off with just this list of things that we don't need to do anymore to generate really, really good genome assemblies. We don't need PacBio data, duplex data, ultra-long, high-C. We don't need cultured cells. We don't need fresh blood. We don't need HERO. We don't need Dorado error correction. These are all amazing techniques, really useful tools, but it's better off if we don't need them. And yeah, so that's it. And I'd just like to thank my fabulous team in Sydney, in particular Hasindu and Igor, who led this work. And yeah, thanks very much for listening.